lot of what I was planning to say has already been said, so you'll have to listen through some of it again, unfortunately, in order for me to be able to go through this, this presentation. But when I started this presentation, I called it Clear as Mud. This was before the two recent approvals of the IMPACT and the FoundationOne assays. And at that point, it was like, where do we go from here? Because you could see a steady stream of changes and improvements over the last year. And I will address some of that in my discussion. What I'd like to do is, I think everyone here knows what precision medicine is. And you've heard a lot about companion and complementary diagnostics. I will bore you a little bit more with some examples and some labeling issues. I will not spend too much time on laboratory developed tests. That's an area that's obviously evolving. You just heard Bill give a lot of discussion about that. So I will skip over that. And also the next generation sequencing tests, I had some information on that. But this is how it is streamlining the regulatory process and how that will emerge and evolve. As you know, the past, we're looking at, this is just precision medicine before, currently kind of shifting in this direction. In the future, obviously the blockbuster drug approach is fast fading. Precision medicine is more targeted oriented, the right drug for the right patient group, but still group focused. Whereas personalized medicine is a more of a network oriented design, getting the information together to give the right drug to the right patient at the right time and dose. That's all I'm going to say about precision medicine. So we're kind of in this kind of in-between phase here. I will not give another description or definition of in vitro diagnostics. I think everyone here after several talks knows what they are. Here's just a few examples for everyone to see. The same thing with companion and complementary diagnostics. Everyone knows that the companion is essential for the safe and effective use of the drug or biologic, and we talked earlier about the number of approved companion diagnostics. But the reason I brought up the complementary definition was we waited a long time before we had a definition about complementary diagnostics. We heard about this test that was called a complementary diagnostic, and just as you've seen in the last couple of weeks, it created this firestorm in device manufacturers' companies. So we received recently, Adam Berger, who from CDRH gave a presentation, and this was the most recent definition that I can find, that it is an IVD that identifies a biomarker-defined subset of patients with a different benefit-risk profile than the broader population, for which a therapeutic product is indicated, but it's not a prerequisite for receiving the drug. So that's the definition that I now use. It's, PD-L1 assays and several other assays have gained this status of complementary diagnostic for good or bad. I won't spend much time on this. It just goes into some of the labeling issues. I did want to make sure that we don't lose sight of the prognostic and standalone assays. A little bit, regulatory would not be any good unless they talked about claims. So I'll talk about, these are the three main claims, and they're not mutually exclusive, by the way. A diagnostic test can be prognostic in one indication, selective in another, or predictive, and sometimes they overlap. So this is very definitive, but it's not always as definitive in practice. But a selection claim will select a patient population eligible for a specific therapy. And again, it's for the companion diagnostic use. Right patient, the appropriate treatment, risk of serious adverse events, et cetera. And then predictive are the ones that are most likely to benefit. And this can be either a companion claim or a complementary claim. And the prognostic claims, which are not tied to drug therapy. I'd just like to give a couple of examples of labels, just so I know a lot of my pharma colleagues ask me about this, so that's why I brought these labels for companion and complementary diagnostics to your attention. And the indication statement is the indication statement for the drug, and ZELBORAF is actually a very good example of a true companion diagnostic. It is a kinase inhibitor indicated for the treatment of patients with unresectable or metastatic melanoma with BRAF V600E mutation as detected by an FDA-approved test. And a lot of my pharma colleagues always ask me, well, why don't they mention the test here? Or why is this in the indication statement? And I think FDA was actually very smart in the way they did this, because that way, as you see today versus then, the technology evolves. So rather than having to update the label every time a new technology evolves for this particular marker, you don't have to go back in and update the label. You just go to the list of cleared and approved companion diagnostic tests, and you should be able to find the test or tests there for your particular therapy. The other aspect of this label that's important is that ZELBORAF is not indicated. It has a limitation of use, not indication for the treatment of patients with wild-type BRAF melanoma. So you definitely have to, and actually it's contraindicated. So you need to have the BRAF V600E status in order to receive the drug. So it is a true companion diagnostic. I brought this example to your attention for a number of reasons, one of which was I wasn't sure why it was not a companion diagnostic originally. It gets back to the discussion about the decisions that are made by the agency and how they make those decisions. Here, this is the drug indication statement. TECENTRIQ is a PD-L1 blocking antibody indicated for the treatment of patients with locally advanced or metastatic urothelial carcinoma. It does not have anything in the statement to indicate that it is an FDA-approved test. The clinical efficacy, however, is described for all patients as well as based on PD-L1 IHC status, SP4 142. No patients are excluded from receiving therapy. So it is a complementary diagnostic. If you go over here, here's the all-patient data. The ORR is 14.8%. You look at the cutoff, which is less than 5%, and you see that the ORR barely is above threshold and, for response. And if you go over here to the actual benefit, the patients that benefit, you can see that it almost is a doubling. When I asked the agency why this was complementary, not a companion, and they said to me the real reason was because this indication had not had any any therapeutic advances in over 30 years, and they did not want to deny patients access to this therapy. So it's more the totality of the discussion and the information that helps to determine whether it is a companion or a complementary diagnostic. I will not talk about the laboratory-developed tests. This guidance has been out since 2014, and you can see that things are evolving, so I don't know what direction that will take going forward. But I did want to just bring to your attention the increased complexity of the assays that we're seeing over the past year. Last year, we saw the FoundationOne Focus CDX BRCA assay approved. That was one marker, one drug, so it was very traditional companion diagnostic, although it was a single-site PMA. Then we saw later in early mid-2017 the Illumina panel that was a 56-gene panel, but again, it was for a single drug for colorectal cancer for panitumumab. And then, of course, we have the Oncomine, which is 23-marker multi-drug assay and for a number of indications. But it also had both analytical as well as companion diagnostic claims. Then, of course, I think everyone has heard a lot today about it. I think you've heard about this maybe once or twice. Of course, we have the Impact NGS panel with the de novo 510K class 2. Again, it is a single site. It is not considered a CDX. And then we have the FoundationOne CDX NGS panel, multi-drug, breakthrough device designation. It's a PMA with analytical and CDX claims. And again, it's a single-site PMA. These are, So we understand that one drug, one test will not be sustainable, but this really advances the discussion. The only thing I wanted to bring up here, it's already been discussed, but that the New York State Department of Health accredited as a third-party reviewer of Vitro Diagnostics by FDA, and the Impact assay was the first submission to go through this third-party review. For the only thing I'd like to point out here is that the FoundationOne test was obviously CDX. It would select cancers as well as an analytical, but it has the CDX claims. It identified 5 cancer types that may benefit from 15 different FDA-approved targeted treatment options. So that's your CDX claim. And the clinical performance of the test was established by comparing the assay to previously approved FDA-approved companion diagnostic tests. This just is the guidance for, this mentioned that it was breakthrough device designation. This is just the guidance that came out fairly recently, and it's there. It actually supersedes the expedited access for pre-market approval in de novo medical devices, and that was issued in April of 2015. This is the newer guidance, and that will, once approved, supersede that guidance, the same concept. So in summary, it was a record year for approval of increasingly complex NGS assays. The FDA has unveiled a streamlined path for the authorization of tumor profiling tests based on the recent 510K clearance of the IMPACT and FoundationOne tests. And here we know that it was a 510K, blah, blah, blah. FoundationOne Breakthrough Therapy Designation Program for PMA approval. And both tests can be used for additional claims in CDX development with proper analytical and clinical validation. So as we had the panel discussion this morning, it sounds like there's still a lot of unanswered questions, and there's still a long way to go. So where do we go from here? So that's basically all I'd like to discuss today. Anybody have any questions?